Perfect. Good morning, everyone. My name is Arjun. I'm a PGY4 neurology resident here at the University of Toronto. Today, I'm going to talk to you about our preliminary work on developing an automated method of sleep detection of movement disorders using deep brain stimulation and machine learning. So first, I'd like to give a huge thank you to my mentor, Dr. Alfonso Fasano, who is the chair of neuromodulation at the Crumble Brain Institute at UHN, here at UHN, as well as Dr. Okianis Fau, who's also been very helpful to this project, and Yosra Hashim, a medical student in Ireland, all three of whom were critical for this project. So to get started, movement disorders are neurological disorders of either fast or slow movements, hyperkinetic or hypokinetic movements. And some examples include tremor, and by the name you can tell, a tremor is a form of regular oscillatory movement around a joint, ataxia, which is a form of incoordination, and Parkinsonism, which we're going to talk all about today. And the most common movement disorders in the world are essential tremor, which is a form of symmetric, usually upper extremity predominant tremor that progresses through age. And the second most common is Parkinson's disease. And in fact, Parkinson's disease is the second most common neurodegenerative condition in general after Alzheimer's, affecting anywhere from 2 to 3% of people above the age of 65. And essentially, to distill it in a very simplified form, the pathology is due to a protein called alpha-synuclein, abnormally forming aggregates in cells and intranuclear inclusions called Lewy bodies, which unfortunately they then deposit in different parts of the brain. And in Parkinson's disease specifically, predominantly in a part of the brain called the substantia nigra, a dopamine-producing part of the midbrain that sends projections to the basal ganglia and is very important for various functions, including movement. And so essentially the end product is a loss of dopamine in the basal ganglia, resulting in the cardinal motor signs of Parkinsonism, which is bradykinesia or slow movements for those who may not have heard of this condition, rigidity or stiffness of movements, resting tremor, and as well as postural instability or gait instability. Now, Parkinson's disease, as I mentioned, has these motor symptoms, which are quite debilitating in themselves, but has a whole set of non-motor symptoms that often start years or decades before the actual motor symptoms begin. Among these are cognitive impairment, autonomic symptoms of the autonomic nervous system being affected. But for the purposes of our talk, really importantly are sleep disorders, such as REM sleep behavior disorder, excessive daytime sleepiness, fatigue, insomnia. And in fact, more than 60% of patients with Parkinson's disease have a sleep disorder. The most classic associated sleep disorder is REM behavior sleep disorder, which is typically during REM sleep, when we dream, we have an inhibition of movements, but unfortunately in REM behavior sleep disorder, there's a disinhibition and there's an acting out of dreams or movements during sleep, as well as insomnia, excessive daytime sleepiness, and restless legs are all associated in Parkinson's disease. And as I mentioned, can precede the motor symptoms by years or decades. And why we care about this is that they have significant effects on mood, cognition, and quality of life, which means that it's very important to recognize and try to treat these disorders. So typically, Parkinson's disease involves the giving back of dopamine or levodopa, which is a precursor of the neurotransmitter dopamine, as the mainstay of treatment for motor as well as the non-motor symptoms of Parkinson's disease. However, there are some adverse effects or necessarily a change in the response over time in Parkinson's disease, where over time, people might have a wearing off of their response, what we call an oscillation, where they have periods of an on response and an off response to the medication. And in fact, over time, levodopa, when it's reached its peak effect, can have what's called drug-induced dyskinesia, which are abnormal involuntary hyperkinetic movements, kind of the opposite of Parkinsonism. And there are also issues with, discontinuous drug delivery of a medication with a short half-life of the medication, issues with gastrointestinal absorption varying, as well as how the drug gets into the blood-brain barrier. And that led to the development of deep brain stimulation around 3.5 to 4 decades ago. And DBS is an evidence-based therapy for patients who have, good response to levodopa, but they start to have these fluctuations in motor response and these dyskinesias with, the peak dose of the medication. And it involves a surgical implantation through stereotactic neurosurgery of electrodes to deep brain structures. And essentially it was found out, many decades ago that high frequency stimulation of specific brain targets can mimic the effect of a lesion without actually destroying the brain tissue. And this stimulation can be modulated in certain ways to get certain effects. And more than 230,000 patients as of 2021 have received DBS, so a significant number of people. And the main targets worldwide are typically the subthalamic nucleus, or STN as we'll call it, and the globus pallidus interna, or GPI, which are the most effective targets for motor symptoms. And it's a very complex decision-making process as to how to choose the targets and what patients are eligible for this therapy. But in short, as you can see from this figure adapted from a review paper, this involves electrodes that are placed in deep brain structures like the GPI you can see in red here and the STN. And the lead of the electrode is actually connected to what's called a pulse generator you can see in blue, which is the form of the computer that actually generates the stimulation paradigm that is then applied at the tip of the electrode in the deep brain. So DBS can also be used for other movement disorders like essential tremor, where the ventral intermediate nucleus, or VIM, of the thalamus can be stimulated for severe tremor patients, as well as in patients with Tourette syndrome-related refractory tics, or a form of movement disorder called dystonia, where the target is the globus pallidus interna, or GPI. Now, in general, neurostimulation has been done classically through an open loop system where, the implantable pulse generator, the controller, as you can see in this figure, provides some form of constant continuous stimulation paradigm, which can be modified, but typically is not modulated over time and remains constant without feedback. However, in the past few years, closed loop systems have begun to be studied, based on the idea that feedback is very imperative to modulating stimulation. And that feedback could be some form of biosignal or biomarker that tells a controller whether or not to change or adapt the stimulation to optimize it. And essentially, one of the ways this could work, or adaptive DBS or ADBS, is the measurement of a CNS or central nervous system biomarker. And one of the biomarkers that are typically studied is what's called a local field potential, which is the summation of hundreds or even thousands of extracellular electrical potentials of local neurons in the brain. And new technology has allowed us not only to stimulate, as you can see by this red lead here, a stimulation lead deep in the brain, but also record adjacent in what are called recording leads, where adjacent leads might be able to record the potentials of nearby brain activity to help give feedback. And the reason this is important is that there has been a correlation between what's called a beta band activity, which is a synchronous activity of deep brain neurons in the basal ganglia around a certain frequency range between 12.5 to 30 hertz, what we call the beta band. And this actually correlates over seconds to minutes with the severity of bradykinesia rigidity, or slowness and stiffness in Parkinsonism. And in fact, treatment-induced reductions in this beta activity correlate with reductions in the motor symptoms, specifically the slowness and stiffness in Parkinsonism. So this is one biomarker that has been studied. And essentially, so far, these type of biomarkers theoretically could be used to feed forward back into some controller, an adaptive DBS system, that then, stimulates, changes the local brain environment, and then those biomarkers of what's actually going on in the brain can be used to feed back to either turn on, turn off, or adjust the biomarkers. And to summarize, for example, on the top of this figure, we see that we might want to, for example, stimulate if a certain type of biomarker, for example, a local field potential activity of a certain type is detected, and instead put the DBS off if that's no longer present, to save battery or to prevent adverse effects of overstimulation. And also other paradigms involve, stimulating if many different states have been sensed. So, 1, 2, or even 3 different states might trigger 1, 2, or 3 different DBS programs. And the reason this is important is this, might be able to optimize DBS. And the reason this might be able to be possible is through recording a biosignal. So essentially recording, whether that's at the electrode or elsewhere, some form of a biosignal, sensing that, and then updating the stimulation based on what the brain is doing. And as I mentioned, the beta amplitude has been used as a sensing CNS biomarker for adaptive DBS that correlates with the motor symptoms, but other biomarkers are possible as well, such as cortical recordings over the motor cortex, where brain activity that synchronizes in a higher frequency called a gamma activity might actually correlate with the dyskinesias or abnormal hyperkinetic movement seen in Parkinson's disease, and can help to modulate DBS to help prevent that. And one of the technologies that allows us these sensing is what's called the Medtronic Percept neurostimulator, which allows live sensing or streaming of local field potentials at the tip of the electrode, but also recording these later on for offline analysis. And in summary, the reason that this is important is that sensing DBS electrodes provide a window into the brain to study different brain states and behavior. and in our case, importantly, including sleep. So they provide a window to the brain, and to revisit sleep, subthalamic nucleus, or STN-DBS, has been shown to improve nocturnal sleep and daytime wakefulness, and that unique basal ganglia LFP changes have been shown in electrophysiological studies to occur during sleep in Parkinson's disease in various parts of the deep brain structures. And why this might be important is that automated detection of sleep might allow monitoring of sleep quality, diagnosing sleep disorders like the ones I mentioned, and potentially farther in the future might guide adaptive DBS to directly treat sleep disorders, which is yet to be developed, or even just simply titrate DBS to help treat sleep at nighttime. So machine learning has been used to classify sleep stage and movement disorders using intracranial recordings, where these recordings data have been labeled based on their sleep stage with what's called a polysomnography test or a sleep study. Now, this is limited to a lab setting over single overnight studies where patients stay in a lab overnight, and a sleep neurologist, will then assess, what are similar to EEG recordings of the brain that tell us what stage of sleep or wake the patient is in, and then label the intracranial recordings that we see in the deep brain as such, corresponding to what sleep state or wake state. But these require broadband, memory-intensive recordings, not feasible over days to weeks. And, these are not really feasible for chronic recordings, capable during real-world activities outside of labs. So far, only lab-based overnight studies have been done. So our goal was to compare wake and sleep state, deep brain electrophysiology over multiple days and nights in a natural home environment, and essentially to try to develop a CNS biomarker for predicting wake-sleep status, and doing this by developing a machine learning algorithm to predict wake and sleep status from intracranial deep brain LFP recordings. And we did this in patients with Parkinson's disease, as well as patients with essential tremor and Tourette syndrome, to provide some more insight in various other diseases and brain targets and how sleep changes, and to compare that to Parkinson's disease. Now, we studied 8 subjects in total that had these Medtronic percept sensing capable DBS that I mentioned implanted. 5 of these with Parkinson's disease, 3 of them having bilateral subthalamic nucleus DBS, 2 of them with bilateral globus pallidus interna DBS. We also studied 2 essential tremor patients with thalamic stimulation in the VIM nucleus of the thalamus, and 1 tic Tourette syndrome refractory tic disorder patient with bilateral globus pallidus interna stimulation. And we recorded these patients over 6 to 8 days, which I'll elaborate in a few minutes, or 5 to 7 nights per patient as they went home over the course of last summer. So firstly, we acquired the local field potentials by using specific software available in the Medtronic Percept system that allows us to, every 10 minutes, record the average activity of the local field potentials of the prior 10 minutes. The caveat being that we could only choose a specific frequency band of activity to measure. So essentially what happens is the DBS electrodes, as a patient is home, are always sensing at some rate, in this case 250 hertz, and the raw local field potential data is then converted to the frequency domain using methods like fast Fourier transforms. And the power within a specific band of interest within a 5 hertz band is measured and then stored. And over 10 minutes, the average is then recorded to the device for offline analysis, stored in the computer of the DBS, the implantable pulse generator. And the way we chose the actual frequencies of interest was to detect, frequency power peaks within the band of interest. And that depended on the patient. So Parkinson's disease patients, as we mentioned, their Parkinsonism or motor symptoms correlate with the beta range. And so we decided on the more affected side to record at the tip of their DBS electrode within the beta range of activity. And on the less affected side, the alpha range, to provide a bit more of a broader scope of frequencies for our recording. For essential tremor, we, because of technical constraints, we were only able to record unilateral recordings, but we recorded in the alpha band as there are associations with that and tremor. And in patients with Tourette syndrome, the one patient, we recorded both the alpha and the beta band. At the same time, how do we label the data that we see as the patients are home? The LFP data, we have it, but how do we determine whether the patient is asleep or awake to label that data? The way we did that was through actigraph wearables using next generation Fitbit Inspire 2 devices. These are devices that were worn every night at the beginning of sleep, and then after the patient woke up in the morning later in a non-lab setting at home. And it works based on heart rate and movement changes. And they actually have, based on some smaller studies, similar accuracy to polysomnography only in certain ways, in certain metrics, meaning the onset and the ending of sleep time, the total sleep time, and the efficiency, but there is no evidence regarding sleep stage accuracy, and they're unlikely to provide any real understanding of what sleep stage the patient is in, as that requires a much more nuanced understanding of what's going on in the cortex of the brain. But nevertheless, there's good evidence that the sleep onset and end time, which is what we cared about in the study, could be measured. We also measured sleep scales that the patient filled out through questionnaires to tell us about their subjective quality of sleep, which I won't go into for the purposes of this talk. And essentially, as I mentioned, each patient went home for 6 to 8 days or 5 to 7 nights, which corresponded to about 900 to 1100 data points within the brain being recorded. As I mentioned, every 10 minutes was a data point. So that corresponded to the total of 5 to 7 nights. And in total, 53 sleep and 61 wake epochs were recorded over the 8 patients. And the data was then exported, in the case of LFPs, to JSON files and the actigraph data to CSV files for offline analysis in Python. So this is an example of how the data looks for an example Parkinson's disease patient, how their beta power fluctuates throughout wake and sleep. The wake being in blue and sleep in orange, labeled from the actigraph recordings. And this data is normalized between 0 and 1 to allow comparison. And you can see that the beta power does somewhat fluctuate, but there's significant overlap in the LFP power values throughout sleep and wake. So each time point, as I mentioned, was labeled based on their wake sleep status from the actigraph recording. We removed outliers and then normalized the data from 0 to 1. And then we actually split the per patient the data into training data and testing data. The first 2/3 of the data being a training set, and the last 1/3 being a testing set. So that then led us to think what would be good features to help us try to predict sleep and wake. As I mentioned, LFP power oscillates through wake and sleep, and there's significant overlap between both states of wake and sleep. And so single LFP power values were likely not useful alone. So instead, we had the idea to use a vector of LFP power values at some given time t to use some sequence of n prior time point LFP values to provide some element of trend as features for the data. Of course, without the actual wake sleep label, just the actual LFP power value. And we did this for using alpha alone as features, beta alone as features, or both alpha and beta for those that had alpha and beta recorded. You can see here, for example, if we were to use 80 prior time points, for example, the 80th time point, as you can see in my cursor, would have not only the current beta and alpha at t equals 80, but the prior beta and alpha for the 80 prior time points. And this would be a one-dimensional vector as our features that define a given time point. So putting that all together, we have our train and test set, but if we were to, for example, use n equals 80 time points, that means that these areas in red could not be used as training data. Of course, we don't have enough prior features to define a given time point before for example, the 80th time point, if we were to use 80 prior time points. And similarly for the testing set, in order to prevent overlap between the training and testing, we could not use this area of second red bar in the testing set. And if this were to be implemented online, it would, approximately correspond to waiting 13 hours before enough features have been recorded to allow classification to happen actively online, which is what we were trying to mimic. So the first step was to do some unsupervised clustering to see how the data clustered. We did this using something called TSNE, which is a method that reduces high dimensional data sets for visualization in 2 dimensions. And it's especially thought to be good for oscillatory data like ours. We then applied on this data what's called HDB scan, a form of unsupervised machine learning, to try to see if, without providing any labels, an unsupervised machine learning method could take these post-TSNE cluster data and see if it could actually label the data somewhat accurately. So for example, if we were to use 5 prior time points as the features of some given point t, we used also the prior 5 LFP values, we see that there's very poor clustering that doesn't provide enough information to discriminate between wake and sleep. With n equals 35, we see a bit more clustering, but with n equals 80, we see that there are much more distinct clusters of sleep and wake that are present and might be more easily separatable. And then applying HDB scan on each of these data that you can see, we see that with just using n equals 5 prior time points, really taking that raw unlabeled data, HDB scan is not able to accurately label the data at all, kind of 50-50, but had an F1 score of 0.75 for seeing n equals 80. So even without a supervised learning method, felt like we were getting somewhere with using 80 prior features. So that then led to supervised machine learning. And because the constraints of the technology, we can only run on low memory devices, the implantable pulse generator, which requires much less memory intensive methods. So we decided to use a random forest classifiers personalized to each patient, which is an ensemble method of learning of constructing decision trees and then using averaging to improve the accuracy and reduce overfitting. We used 100 estimators and a max depth of 10 trees. And again, this we felt was much easier to train on a device than say neural networks, which would not be possible to implement on our device if we wanted to do this online, for time series analysis, and then, would have less overfitting than decision trees and easier to train on smaller data sets. So essentially, to just go back very quickly, our problem is really one of not actually predicting, typical time series, analysis where we're trying to figure out the value of the time point, trying to predict how the trend of data is, but rather simply the label of whether the patient's in wake or sleep. So a different sort of task than usual time series analysis. So coming back to what we did, as we saw for the prior kind of logic why we used 80 prior time points, we decided to use 80 prior LFP time points as features for patients that had bilateral sensing, which were the 4 out of the 5 Parkinson's disease patients. We studied classifiers trained based on both alpha and beta powers. And we also individually trained on just alpha or beta alone for patients with bilateral sensing, just to compare how that might stack against alpha and beta as features. We also, for patients that had either just beta or just alpha, used those features alone. And for all of the above, we also applied separately an averaging method where every 3 preceding or 5 preceding time points were averaged together to help reduce noise and fluctuations, and then use those as input features and retrain the classifier. So in general, this is the schematic of using both alpha and beta, alpha or just beta alone, with or without averaging, training our machine learning model, and then seeing what the performance was. So very quickly, just comparing the statistics between wake and sleep across various diseases and deep brain targets, we did show that there were some changes that happened in the basal ganglia, in STN, the GPI, the VIM, across Parkinson's disease, essential tremor, and Tourette syndrome, which I won't go into here, but really kind of did confirm prior findings on electrophysiological studies of how the local field potentials do change in wake and sleep. So this here is our performance. So using both alpha and beta as features, we had an aggregate over, kind of combining the confusion matrices of all 4 patients, an accuracy of about 85%, a moderately accurate model with an F1 score of 0.84 with macro averaging. And we had a maximum accuracy of 88% and F1 score of 0.89. But as you can see, using alpha alone, we had similar, maybe slightly higher accuracy in predicting wake and sleep with a maximum accuracy of 92% and an F1 score of 0.91 and an F1 score and accuracy of 0.86. And very similarly for using beta alone as well. And you can see here some of the confusion matrices. And between averaging or not averaging, we essentially got very similar results. So averaging didn't seem to provide a significant benefit in classification performance. For patients with essential tremor, as you can see here, we had a maximum accuracy of 0.86, but averaged over, or rather compounded over the 2 essential tremor patients. The combined confusion matrix had an accuracy of 0.83 or an F1 score of 0.82. With Tourette syndrome, the 1 patient we saw using alpha and beta alone provided an accuracy of about 89%. Using alpha alone was similar, but beta alone provided very poor relative accuracy of about 0.68. So there were some significant limitations to our preliminary study. So we used about 900 to 1,100 data points, which was, high per patient, but limited by the small cohort of patients. And just having 8 patients alone meant that we couldn't aggregate all of these various patients and, train on many patients together and then test on patients using an aggregate of many patients' recordings put together. We also only use narrowband LFP recordings using just 1 to 2 specific frequency bands. As I mentioned, there's significantly more information in the raw recordings, which have all of the sort of frequencies that are represented in the raw neuronal recordings. And there's a potential significant increase in classification performance without the bias of frequency band selection. However, that requires significant memory, and that prevents long-term recordings over many days and can only really be done over the course of one night feasibly. So in addition to that, we used actigraphs, to measure when the patient was in sleep or not, to label our data. Sleep studies are the gold standard, and they require overnight, multi-electrode kind of EEG leads put on the patient's head to determine accurate sleep state, which is a gold standard, but requires a lab, which doesn't allow sort of chronic recordings over many days. And using actigraphs alone, as I mentioned, we could definitely not actually tell what stage of sleep the patient is in. That's very important because some disorders like REM behavior sleep disorder happen preferentially during REM sleep, and having an ability to actually see what stage of sleep the patient is in is extremely important. And so that's something that we were not able to do in this study with the limitations of our technology. So that leads to the next step of future studies to, potentially use different frequency bands to improve sleep detection. As we know, the cortex slows down significantly during deeper stages of sleep into the theta and delta range, or much slower frequencies. So our goal for next steps that we hope to do in the near future would be to take some patients and record them overnight with a formal sleep study to identify the deep brain recording bands that are of interest, not simply the alpha and beta that we chose, but are there other bands that might be important? And repeat this study of chronic narrowband recording using these optimal frequency bands to simply classify wake and sleep and see if we can improve from this kind of moderate accuracy that we see now. In addition, we'd also want to train a multi-class classifier to not just predict sleep and wake, but actual sleep stage of awake, deep sleep, light sleep, and REM, these various important sleep stages. And the way we'd be able to do that would be to use other forms of technologies like ambulatory sleep monitoring devices or ambulatory EEG or some similar technology that might allow us to actually, offline, then label the data much more accurately based on sleep stage, and then retrain classifiers for a multi-class problem. And so in addition, this capability simply to detect the wake sleep status might provide insight into sleep patterns. And so other studies to just simply send patients home and correlate that to their quality of sleep and duration of sleep would be important using similar methods, and possibly even target treatment of sleep disorders using DBS. And as I mentioned, adaptive DBS so far has been, programmed so as to identify, biosignals that correlate with motor symptoms, but sometimes theoretically, this might apply stimulation inappropriately during sleep but not desired. So having a sleep-aware adaptive stimulation system would be, more beneficial. And really, the goal of the future would be to have increased computational abilities on these implantable pulse generators to allow chronic raw LFP recordings, not limited to specific bands, but allowing us to record over many days or weeks the actual raw recordings, as well as potentially allow deep learning online, not limited to less memory intensive methods that we currently are limited to. And finally, our final goal would be to actually create an adaptive DBS system to directly treat sleep disorders. That would be, the end goal of these preliminary work. So these are my references. I'm happy to share them with you later on offline. And thank you so much. And again, special thanks to Dr. Fasano, Dr. Val, and Yosar, without whom this project would not be possible. Thank you so much for your attention.
Thank you so much, Arjun, for the talk. It was great. Maybe we can move on to questions now. I'll start off with Dr. April Kademi, if you have any questions.
Sure. Thank you so much for the nice presentation, Arjun. I have a question about the data that you collected, the LFP.
Sure.
So when you show, can you actually bring up the one where you were trying to do the training and testing?
Yes, let me see if I can go back in my slides here. So would this be the one that
exactly?
Okay.
So this is a recording in the beta and different frequency ranges, right? You did alpha and beta.
Right. So this is just the beta shown. You're right.
Yeah. So like when I look at it, when I look at your data, it does, I do see the cyclical nature in it, right? And which is what you were saying that one of the projections, one of the unsupervised projections kind of also showed that as well. But do you think that, the cyclical nature of the data, like it seems like there's a drift in the data. Do you know why that is?
I'm not sure, but patients that have, Parkinsonism, the beta band activity correlates with the motor symptoms. So it could be that at various stages they had different levels of, disease response to their treatment, and that fluctuates over days and weeks. So you can see a natural change over days to weeks, and that might provide a complication in terms of the baseline kind of shifting because during the wake state, their Parkinsonism is different, and that could correspond to why the beta fluctuates so much during daytime between, say, the beginning of the 5 days and the ending. That's what I would probably attribute that to.
Yeah, one of the things I was going to suggest is to maybe just try to Z-score normalize your data.
Okay.
Because it could be just a sensor bias or something changing over time, but not really related to what you're trying to measure.
Okay.
And so if you do a mean and standard deviation, subtract the mean, divide by standard deviation, you might be able to normalize your data a little bit more.
Right.
But yeah, that was my only question. Thank you very much. Great
Yeah,
talk.
it would be great to talk offline about that as well.
Sure.
So maybe I'll contact you then. Thank you.
Sure. Thanks.
Thank you, Dr. Kademi. Any other questions?
Yeah, quick question. So I like how you visualized the clusters, all that stuff. So my question is, TSNE is really great for us humans to visualize what's going on with the data. Why did you input the 2D kind of reduced features to your unsupervised learning algorithm instead of feeding the 80 features directly there?
Yeah, I guess I don't have a good explanation as to that. It was just that for us to visualize the data, we reduced it to 2 dimensions and in a sort of a different feature space. So we thought that potentially that would allow us to visually compare those by applying the labels with HDV scan and comparing how that looks in that figure, if I can show you again. But I guess that's something we could do as well with all of the 80, but just a bit hard to visualize. Although we would be able to get, an F1 score and, performance from doing that. Maybe we won't be able to visualize it, but we could still do that. Yeah.
Thanks.
For sure. Yeah, maybe that's something that we can look into. I think Dr. Murray has his hand up. Hi, Dr. Murray.
Hey, great presentation. Really cool. I was wondering what the limitations are on the device for collecting samples. What are the realistic sampling techniques? Like, can you sample multiple frequencies, or do you just get a raw signal and you process it later? Or
yeah,
how does it work?
It's a great question. So I'll just quickly go back to here of how we went ahead and did that. So overnight, essentially, you could record the raw data. That does require a lot of battery, but it could be done, I think, every 10 minutes. Unfortunately, the technology requires that a reset in that recording is done. So someone every 10 minutes has to actually, store what has been done for 10 minutes and then reset the recording of the raw data. So it cannot just be streamed long-term. However, what we're able to do long-term is specifically choose a frequency band, which in this case, as I mentioned, we used those bands of interest, but in one electrode, we were able to only do one band, in one side, and the other side, we can do another one. So realistically, we can only do 2 bands chronically with the current technology. However, overnight, we could, and if someone overnight is present to actually store the data, take a raw recording, and, that would be helpful because we could not only just, find the bands of interest, but convert that to like a spectrogram or scaleogram using like a wavelet transform, and then, train some form of deep learning network on that. So, I think unfortunately that's our limitation now. I hope that answers the question that, we can only choose 2 frequency bands maximum. We can't do more than that long-term unless the patient is in somewhere where they're monitored every 10 minutes, for example, overnight, to stream the raw data and record that.
The one thing you might want to do is collect, theta frequency and delta frequency from separate bands, and then cross them, because you'll see in various states they separate out nicely in terms of the delta power versus the theta power, and you could actually track over time how people are moving through these states.
Right.
But the sampling is going to be a problem. The amount of data that you can pull, it sounds like it's a battery drain, that's problematic.
Yeah, but I think for sure what you're mentioning about choosing the sort of like a theta or delta band and then maybe some other band that's quite far away might provide enough of a separation in like the feature space to get a broader understanding of sleep beyond what we just were able to. And I think I'm confident. We can have a discussion like how does sleep actually change in those frequency bands deeper in the brain? Because I know from sleep studies and our understanding of sleep, we talk about the cortex, but what happens deep in the brain is a bit more of a mystery, which is what we were trying to look at here, but not in the theta and delta ranges. So that would be really cool to look at. And maybe that will be helpful because maybe the brain slows down to that as well. There's also sleep spindles that are present, I guess, potentially they come from the thalamus. And so part of why we wanted to study patients with thalamic recordings. But there's so many different aspects of sleep that are over many frequency bands. So as you say, we're limited by the technology, but maybe we can still optimize what we have based on the limitations.
Good point.
Thank you.
I had another quick question actually. I was wondering, you were talking about closer to the end, the gold standard for doing these measurements. What was it again?
Yeah, so that's actually in terms of measuring sleep and telling when a patient is in sleep, what stage of sleep they're in, is actually called a sleep study. If I can quickly bring that up over here. So that's called polysomnography, which Dr. Mary, who was just talking, is a sleep specialist at Sunnybrook. So that essentially involves patients having electrodes on their scalp overnight and measuring their brain activity as they sleep. So we can see exactly what the brain is doing from a cortical perspective. And that can label the data much more accurately than just wake or sleep, but having specific types of sleep. And that can tell us, maybe some specific more information about sleep that we don't get just simply from the actigraph. Yeah.
Just a quick idea, but I don't know if the feasibility of it, but with the, because you were mentioning like the gaps, the 10-minute gaps in the metrics that you're recording, if this much more effective way of looking at sleep, if you're able to train some sort of thing that connects these measures, the 10-minute gap measurements versus this one, if there was some sort of way of connecting those two and then make the predictions that way, would that be useful?
Yeah, that's a great idea. I guess having like a second classifier to I guess connect what we could send patients on chronically, connect that to what we do overnight. So then that can maybe tell us about sleep stage. Yeah, I think that's a great idea. I have to think a bit more deeply about how we do that. So I'd love any ideas that you have about how we do that. And one of the ways we were thinking was an ambulatory system, which we're trying to work with some other sleep specialists to see, how can we measure the actual sleep stage and a bit more accurately too. And, ambulatory devices are being developed and that's something that maybe could bridge the gap beyond just this actigraph. So yeah, I think that's a great idea.
Yeah, maybe we can chat offline then.
Yeah, thank you.
Okay, well, in that case, thank you again, Arjun, for the great talk. It's very insightful.
Thank you so much. Thanks for having me.